So I'm going to give a short overview of adenosine deaminases, so you will get some idea what do they do. I'm trying to answer some questions that you have asked, but because I wasn't on this panel, so I decided just to tell you in my introduction. So, but the idea is that adenosine is quite important signaling molecule, actually. And adenosine deaminases, they eliminate adenosine by converting it to inosine. And actually, the main adenosine comes from ATP, and ATP is produced during the inflammation. Either it's pumped out from the cell, or there are some cells that are actually dying, so they release a lot of ATP. And the concentration of ATP is really high. It's a millimolar range, so it can be hydrolyzed by some enzymes that are located on cell surface of the cells. They're called ectoenzymes. They're called like CD39 or CD73. So they can produce adenosine monophosphate and then adenosine. And adenosine is taken up by adenosine deaminases and converted to inosine. So actually, this picture is coming from this ADA1 deficiency. So then adenosine can bind to 4 different adenosine receptors. They're called A1, A2, A2B, and A3. And these adenosine receptors are also important for the controlling of the cell function. There is also adenosine inside of cells and outside of cells. So we don't know what is the function of these enzymes is more important, whether it's extracellular function of these enzymes or intracellular function of these enzymes. Okay, next. Okay, so to make it more complicated, I would like to say that all these 4 different adenosine receptors, they have different affinity for adenosine. Otherwise, so they bind to adenosine at different concentrations. So let's say adenosine receptors A3, A1, they can be activated at very low concentration of adenosine. And for the activation of A2B and A2A receptor, you need to increase the concentration of adenosine. So that's why these receptors, they work at A1 and 3, they may work in normal physiology, and A2A and A2B, they can be activated at extreme pathology. So moreover, these adenosine receptors, they expressed on all cell types, and the level of the expression is different, and also the combination of these receptors is also different. And besides, they also controlled, the expression level is controlled, so that's why, for instance, in normal physiology, you may get expression of 1, 2 receptors, but in the case of pathology, you get expression of other receptors. So it's quite complicated because, for instance, activation of the same receptor on different type of cells can lead to absolutely different outcomes. So, next. So in general, activation of A1 and A3 receptor leads to decrease of cyclic AMP, which is a very important molecule. And then activation of A2A and A2B receptor leads to increase of cyclic AMP. So there is a balance between the function of this, between the signals that signaling between A1, A3, and A2B and A2A receptor. So it's like a feedback mechanism. So the major drug is actually caffeine. And actually yesterday on the plane I saw a documentary about caffeine. So, and this is the cause of colonization of Africa by British people actually. So I didn't know about this. And so caffeine is a major drug, and it actually binds to adenosine receptors, and it blocks the binding of adenosine. So, and adenosine has many different functions. I'm not including these temperature regulations. That's why probably patients, they have high temperature and fevers. But also, it's also involved in regulation of neural system, immune system, and so on and so on. So it's quite complicated. So here is just an example of different receptors that are involved in different diseases. So actually, you won't be able to find any disease where you don't have any adenosine receptor involved. So what is about this adenosine receptor, or adenosine deaminases? Actually, there are 2 different types of adenosine deaminases, AD1 and AD2. AD1 was studied in the 70s, and it was found that deficiency in AD1 causes severe combined immunodeficiency, and it's about 15% of all cases of severe combined deficiency due to the mutations in AD1. And this enzyme is mainly intracellular, so it's inside of cells, and it acts at normal pH, and it's really active at low concentration of adenosine. So what is about AD2? AD2 is mainly, it's also could be present inside of cells, and actually there is, I think there will be another talk, but we also have found that there is a lot of AD2 inside of monocytes. But besides, AD2 can be found in extracellular space, and if you compare AD2 and AD1, AD2 almost inactive because it has low affinity for adenosine and it works at really high concentration of adenosine. So this is a major difference between these two enzymes. And also the activity of this enzyme is maximum at low pH when you have inflammation or tumor growth. So ADA2 is secreted mainly by macrophages or monocytes or dendritic cells, but as it was shown before, it also could be secreted by B cells, but this should be checked. And also it binds to the cell surface, and we have speculated that it binds to proteoglycans, but there could be also adenosine receptors on other unidentified receptors on the cell surface of cells that bind AD2. As for AD1, it can be found both inside of cells and outside of cells, and outside of cells, AD1 binds to the molecule called CD26, which is expressed mainly on T cells, and also it can associate somehow with adenosine receptors as well. So when I was at high school, so I was developing this assay to measure the activity of different enzymes. Actually, it looks exactly the same as this HPLC assay. So you can actually follow the hydrolysis of AMP to adenosines and to inosine and other products. So using this assay, I have found that actually immunoglobulins that are produced by classical ethanol fractionation, they contain a lot of adenosine deaminase activity. And this adenosine activity was due to AD2. In other words, AD2 was co-purified together with immunoglobulin. So the main problem was to purify this AD2 and isolate it from immunoglobulin, actually. So, but luckily I found that AD2 binds to heparin. So it was possible to isolate a minor fraction of this enzyme, and it took about 15 years to make it because at that time, in 2005, there was a possibility to run mass spec and cut a band from a gel and identify this gene for this enzyme in one day, actually. So, and apparently, the gene for this enzyme is called cat-eye syndrome critical region 1, and this is due to the also rare disease, genetic disorder, which is called cat-eye syndrome. So, but this is due to triplication of chromosome, and it's not clear whether AD2 is really important for this syndrome because there are also 15 other genes on the chromosome. But because the function of other genes is known, so it was supposed to consider that AD2 is probably important for the development. So essentially, in this case, you have more AD2 activities than normal. So, and it was found that actually AD2 is different from AD1. It's actually bigger. And it also has homologs in flies that were identified in 2001. So next, it was possible just to see where this gene is expressed. And it was clear that the gene was expressed only in immune cells. And the so it's also present in T cells, CD8 cells, B cells as well, NK cells. But the majority of this enzyme is present actually in monocytes and myeloid cells. So actually what I have done, I just cloned the gene from a monocytic cell line, and it was overexpressed in insect cells. So it's actually nicely expressed. And these cells, they also have a homolog for the ADA2. So then this enzyme I have crystallized, and with the help of my brother, he is a crystallographer, so we got the crystal structure of AD2. So this is the crystal structure of AD2 with its inhibitor, It's called it's a pentostatin. It's also a drug to cure leukemia, and it binds both to AD1 and AD2. So the structure of AD2 is quite nice. There are two, it's a dimer protein, and we suppose that these parts are required for the binding to a known receptor, and then another part is required for the binding of proteoglycans. Okay, so then it's also the difference between a catalytic region between AD1 and AD2, and there is some extra space for adenosine. That's why adenosine doesn't bind well to AD2, but it's still possible to use this extra space to design a specific inhibitor for AD2. This is what we are doing. So then I have analyzed cells from AD2 deficient patients from the NH, and it was found that actually in 8 patients out of 9, if you can compare NK cells and NKT cells, these are NK cells, NKT cells, you can see clearly that AD2 patients, they don't have NKT and NK cells. So, and also it was found that CD16 positive monocyte subset is missing in AD2 patients, deficient patients. As for B cells, besides what was discussed before, we found that actually CD25 positive cells, the cells that bind interleukin-2, they also missing in AD2 deficient patients. And if you compare to other patients with autoimmune diseases, these are clearly difference between AD2 deficient patients and other patients. Okay, so what was found recently is actually that AD2 and AD1, they bind to different cells. So essentially, if you look here, it's not clearly seen. So AD2 binds to neutrophils. It also binds to monocytes, B cells, NK cells, and NKT cells, CD8 and CD4 positive T cells, they do not bind AD2. But in the same time, as I talked, CD26 molecule is a receptor for AD1. It's expressed exactly on NKT and T cells. So essentially, AD1 and AD2, they bind to different subset of cells. And to go further, we could find that even in the subsets of NK cells that bind better AD2, so this is NK cells, they are present here. So let's say CD56 high cells, they bind less AD2 than, let's say, CD56 positive cells. And the same goes for monocytes. CD16 positive monocytes, they bind better AD2. And the CD16 negative monocytes, they don't bind AD2 as well. But in the same time, AD1, we found bind to the CD16 negative subset. So essentially, and then also we found that the cells that normally stain for the regulatory T cells, they also bind AD2, and AD1 doesn't bind to regulatory T cells. So to sum up, I can say that, okay, so CD16 monocytes, they actually have a lot of AD2 inside, and they can secrete AD2, and we don't know what is the condition for the secretion. And then AD2 can bind either to regulatory T cells or neutrophils or B cells and NK cells. Then AD1 is mainly present inside of cells, but it also can be found on the surface of effector T cells, and it may also participate in bridging of myeloid cells and, let's say, dendritic cells and T cells, but it's not proved yet. And we actually don't know exactly what is the receptor for AD1 or AD2 on monocytes. It could be either adenosine receptor or it could be some other receptors. Okay, so but that's why actually you may have such a different phenotype, because there are many cells affected if the function of AD2 extracellular is important. Okay, so here we can also show that AD2 deficient patients, they downregulate the expression of this receptor, because here I just compared this a control and this a patient 7, and you can see that the level of expression of on all cells of this unknown receptor is downregulated. So this is the main goal. So what we want to do is to find the receptor for AD2 and AD1, of course. And once we know the receptor, we probably know the function of these enzymes. Then we would like to continue working on the mechanism of the regulation of activity by adenosine deaminases. Then we want to make a specific inhibitor for AD2 because it's absent now. Then we'd like to continue developing kits for diagnostic and treatment of immune disease disorders, including AD2 deficiency. And then we want to continue with studying structure evolution of AD2 because now we have AD2 from bacteria to humans. So that's why we can crystallize AD2 from different, from mammalian compared to bacterial and from flies, from frogs, and so on. That's it.